So we're still waiting a few more minutes. We'll get started around 12:03 to give people a few more moments to sign up.
Recording in progress.
All right, so let's get started. I'm really excited to welcome everyone to the cancer session of the Leading Edge Symposium. I think today we're very lucky to be able to hear from 4 really talented scientists in the cancer space. And so I know that we'll be hearing a lot of exciting science today. So just some notes. So if you have questions after the talks, feel free to either raise your hands and ask them yourselves, or you can put them in the chat and I'm happy to read them. And then also speakers, just try to be cognizant of the time and stay on time as much as you can. So let's just dive right in and get started with the first speaker. So I'm very happy to introduce Sarah Naomi Olsen as our first speaker today. So she's coming to us from the Dana-Farber Cancer Institute, and has done a lot of exciting work studying transcription factor biology and chromatin complexes, and has some background in studying like leukemias as well as breast cancer. She's also a Damon Runyon Fellow, and so I'm very excited to hear your presentation today, Sarah.
I already failed with the simplest task after 2 years of Zoom. So there you go. Hold on. Let's try this again. Cool. Thanks for the introduction, Lindsay. Yeah, as Lindsay said, I actually go by Naomi, which is why Sarah's in parentheses. But it is confusing because Sarah is my actual first legal name and Naomi is my middle name. But anyways, I go by Naomi, and I'm super excited to talk to you guys today about my project and just generally my interest in mechanisms of transcriptional regulation by chromatin complexes, specifically in the context of cancer. And so epigenetics obviously does not need much of an introduction. So just very briefly, as you all know, DNA is wrapped very tightly around these histone proteins. And so specific modifications either on the histone tails or on DNA itself can regulate transcription and can determine which genes are on or off at any given point. And that is obviously incredibly important during development, but also more recently has really been recognized as a crucial and emerging hallmark of cancer. And so in the field, we typically think of 3 classes of proteins. There's the writer proteins, which, who as the name suggests, write epigenetic marks, so they will deposit these modifications on either histone tails or DNA. Conversely, there's a class of proteins called erasers, which will remove those marks. And then lastly, there's a class of reader proteins, which will recognize those marks and then either recruit transcriptional activators or repressors. And mutations or alterations really in any of these different groups of proteins have been reported across many different tumor types. But my project and the focus of my lab really is one of these writer proteins, specifically MLL1, or also called KMT2A. And so MLL1, as you can see over here, is a very large protein with many different domains. And the catalytic H3K4 methylation domain is at the very C terminus. But in cancer, specifically in leukemia, MLL is quite frequently chromosomally translocated at this breakpoint region. And so MLL then is fused to a variety of different C-terminal fusion partners as shown down here. But as you'll also notice is that this breakpoint region really occurs prior to the catalytic set domain. And so these MLL fusion proteins are thought to be oncogenic, not through aberrant H3K4 trimethylation, but rather they're thought to activate genes through recruitment of alternative binding partners to chromatin. And so MLL-AF9, for example, which is the fusion protein we most heavily focus on in the Armstrong lab, it's actually the most common fusion protein in pediatric leukemia. And so MLL-AF9, as shown up here, can directly recruit the lysine methyltransferase DOT1L, and DOT1L then can methylate H3K79, thus leading to chromatin decondensation and facilitating transcription of genes. Alternatively, MLA9 has also been shown to directly recruit the super elongation complex, mediating productive transcriptional elongation. And so it's really through recruitment of these alternative binding partners that these fusion proteins are thought to drive or aberrantly drive gene expression to cause cancer. And so in the last 2 decades, we've really made a lot of progress in terms of targeting these oncogenic fusion protein complexes. We have developed catalytic inhibitors to DOT1L that have been promising in leukemia and have been explored in clinical trials. More recently, there's also been a big emphasis and drive to develop inhibitors of the menin and MLL interaction, and these are currently actively being investigated in clinical trials. And then in addition to these more targeted therapeutic approaches, there's of course a plethora of inhibitors that really target the transcriptional machinery more generally. But despite kind of the progress we've made with developing all these inhibitors, we don't yet know what the effects are of direct targeting of the oncogenic fusion protein. And so when I started my postdoctoral project, I wanted to ask what would be the effects of MLL-AF9 inactivation, both on the leukemic chromatin architecture, as well as on MLL-AF9 driven transcription. And then secondarily, I wanted to know how would this gold standard now of direct MLL-AF9 inactivation, how does that compare to some of the currently available small molecule inhibitors that we have? And so to do that, I made use of the protein degradation approach. Some of you might be familiar with this technique, but basically your protein of interest, in my case MLL-AF9, is fused to an FKBP12 tag. And then upon treatment with these bifunctional DTAG molecules, your protein of interest gets recruited to the E3 ligase complex and gets ubiquitinated. And that is because these DTAG molecules are able to, on the one hand, directly bind the FKBP12 tag, and then on the other side, they're able to directly bind the E3 ligase complex. And so through bringing your protein of interest into close proximity with the E3 ligase complex, your protein of interest will get ubiquitinated and then ultimately proteasomally degraded. And what's really unique and special about this approach is that this proteasomal degradation occurs typically within minutes or hours of DTAG treatment. So the kinetics are such that you can really very acutely assess cause and effect relationships. And so I made use of this approach and tagged my MLL-AF9 protein. And you can see here by Western blot that as early as 15 minutes, we see quite significant reductions in MLL-AF9 protein level. And then at 30 minutes, it's virtually gone. And so we wanted to use the system to really clearly define MLL-AF9 chromatin occupancy. And so over here now you're looking at a tornado plot, and you can see that in our DMSO treated condition, we get very nice MLL-AF9 signal. And then up on DTAG treatment, that signal is completely ablated, indicating that the signal we get over here is actually specific. And so we then rank ordered the signal around the transcriptional start site. And we found that there is a subset of genes, as shown here in black, around 200 or so, that had a very high load of MLL-AF9 around their promoters. And those genes we've then defined to be the MLL-AF9 target genes. And so now using the system and using the protein degradation approach, we wanted to know what happens to these MLL-AF9 target genes up on MLL-AF9 degradation. What are kind of the most immediate effects? And so to To get at that, we used a technique called pro-sequencing or pro-seq. It's a technique that really allows you to map transcription genome-wide and to look at actively transcribing or engaged RNA pool 2. And so what we found was that there is a subset of MLA9 target genes where even at 15 minutes, we saw very strong defects in transcription. And so what you're looking at here is the pro-seq read density plotted along the full length of the gene body. And what you'll notice is that at 15 or 30 minutes, as well as 3 hours, we see significant reductions in transcription or pro-seq read density along the full length of the gene body. And that is particularly pronounced at this subset of MLA9 target genes. We do also see reductions at all other MLA9 target genes, but as you'll notice, they're not nearly as strong or robust as at the smaller subset. And then, of course, as a nice kind of sanity check at our non-MLA9 bound genes, we saw no effects on transcription. And so I'm not showing you this data here, but we also actually assessed the chromatin state, and we wanted to know if MLA9 degradation had effects on the chromatin state. And we found that yes, it did, but these chromatin state changes occurred subsequent to these more immediate transcriptional changes. And so this work really allowed us to conclude that MLA9 directly promotes and drives transcription. And so now with this knowledge at hand, we wanted to go back and ask, how does that now compare to treatment with some of these small molecule inhibitors, specifically an inhibitor of the menin-MLL interaction or a .1L inhibitor? And so again, now we began by looking at ChIP-seq, and what you'll see immediately is that treatment with these inhibitors led to modest reductions of MLL-AF9 genome-wide, but by no means as effective as DTAC treatment. And that you can also see that over here in these ChIP tracks. And I also want to draw your attention to that we saw some loci where treatment with these inhibitors had actually absolutely no effect on MLA9 occupancy. And those loci tended to be relatively resistant to these inhibitors. And so this then, of course, raises the question of what about combination treatment? Can we completely ablate MLL-AF9 from chromatin when we combine these two drugs? And indeed, that is what we found. Again, over here, we're looking at tornado plots, and what you can see immediately is that the combination completely ablates MLA9 from chromatin, very similar to DTAC treatment. And even at those loci that I mentioned that were relatively resistant to single agent treatment in terms of MLA9 occupancy, even at those loci did the combination completely ablate MLA9 signal. And so we published this work, of which a much more detailed kinetic analysis, of course, of MLA9 degradation, as well as a much more detailed comparison of degradation to treatment with these inhibitors. And we published this work earlier this year. But kind of what I want to do next and where I want to go from here is that I really want to use this insight and some of these tools that I've now gathered throughout this project to ask very specific and mechanistic questions in breast cancer. Because breast cancer, despite all our advances that we've made, metastatic and recurrent disease continue to present a major clinical challenge. And so particularly because when the disease recurs, these breast cancer cells tend to be quite heterogeneous in nature. And so some of the more targeted therapies usually aren't as effective as we would like them to be. And so I think that targeting chromatin complexes and targeting transcription would present a very nice therapeutic strategy in that context. And so as Lindsay already alluded to, for me, that is really actually kind of coming full circle, or rather going back to my roots, where in graduate school, I actually studied breast cancer. Specifically, I looked at a BAR RAS pathway activation in breast cancer and how that contributes to breast cancer recurrence and metastasis. And so now I think I'm kind of in a unique position, or in a great position, to really combine my expertise in breast cancer from graduate school with some of these more epigenetic and mechanistic tools that I've gained in my postdoc, to really combine the two and look at some of these epigenetic regulators as new therapeutic strategies in breast tumors. And so just very briefly now at the end, as some of these complexes that I've been showing you throughout, like menin and MLL and .1L, those same complexes actually also are dependencies in a subset of breast cancer cell lines. I'm showing you down here just the broad data set, just CRISPR depletion data set. And so what I want to look at is I really want to explore these complexes and really biochemically characterize the composition of these complexes in breast cancer. And then, of course, also look at chromatin state and transcriptional changes, particularly in response to some of these small molecule inhibitors that I've been talking about. And then lastly, I don't have time to show you this data, but we do know, and we do have some evidence that shows that these complexes cooperate with estrogen receptor-driven transcription in breast cancer. And so I'm also currently actively investigating the role of these complexes in endocrine resistance and resistance to some of these estrogen receptor targeted therapies in breast cancer. And with that, I want to thank everyone. I want to thank, of course, my mentor, Scott, as well as my funding, and then also Leading Edge for giving me a chance to talk today. And it's been very exciting. I've only been a fellow for a couple months, but it's already been great so far and really enjoyable. And thank you for letting me share my data today.
Thank you very much, Naomi. That was a really beautiful talk. So if you have any questions, either put them in the chat or raise your hand, but I wanted to get started with one. So I thought you made a very interesting point. You said you didn't have much time to show data about the transcriptional changes happening before these chromatin changes. And so I guess my question's a little sort of two-part. First, by using this degrading approach, were you able to sort of tease apart those primary and secondary effects that maybe people have with other methods weren't able to? And then what are the implications for that for the therapies? So are you seeing that some of these impacts or effects are more by these primary effects, and then the chromatin rehab modeling that happens secondarily? Is there some considerations there for the therapies?
Yeah, you really hit the nail on the head there. So yes, I think that that's a fantastic thing of the protein degradation approach, that the kinetics are such that you can get a little bit of cause and effect like that. And so I didn't have time to show the data today, but we saw these transcriptional effects as early as 15-30 minutes. But some of these chromatin changes, as you know, H3K27 acetyl or H3K79 dimethyl, as well as other, histone marks, or also ATAC data looking at accessibility. Those tended to occur later, like at 3 hours, 24 hours. And so in the paper, we really hypothesized that these immediate transcriptional effects occur, and then the chromatin changes are more there to solidify these effects, but they don't drive them from the beginning. And so in terms of how that looks with the inhibitors, is that we're currently it's a little bit problematic, because the kinetics of the inhibitors are such that you can't, since it doesn't happen within minutes and hours, we can't really get the timing right to get that window where we could get that. So we are currently in the process of, either endogenously or exogenously tagging some of these menin and .1L, and then using the DTAG system to degrade those and then get at it kind of that way, if that makes sense.
Yeah, makes a lot of sense. All right, we have a question from Richard White.
Hey, that was really cool. So just a question. So do you think during metastasis, do you think that there's sort of a different complex of chromatin factors and transcription factors that get affected by the local microenvironment, such that the kinetics, especially of like inhibitors, would be vastly different in, let's say, liver versus lung versus brain or something like that?
Yeah, I think that's a very good point. And that is something that, in all honesty, the Armstrong lab, at least, is not usually thinking about, because they're thinking about hematological disease, where it's obviously very different than if you're talking about solid tumors and metastatic niches there. So that is something that I would have to, so the goal now for me looking at some of these complexes in breast cancer is to get at exactly those types of questions. And that would be something that I would be looking at. Those compounds, they are, as I mentioned, in clinical trials currently, but of course only in leukemia settings. And so the goal would really be to lay the foundation of also looking at those compounds in potentially some of the solid tumors and getting the kind of preclinical data to start those considerations. So I don't long way of saying is I don't have an answer for you, but
Yeah.
it's a very good question.
It'd be super interesting if different sites respond in different ways in those clinical trials. So anyway, great talk.
Exactly. Thank you.
All right, well, thank you so much, Naomi, again, for a really beautiful talk. In the interest of time, we're going to move to the next speaker. So our next speaker is Miranda Hunter, and she is joining us from Memorial Sloan Kettering Cancer Center. So Miranda has done a lot of exciting work trying to understand melanoma invasion using zebrafish, and she's going to tell us a bit today about some of the mechanisms, including some spatial, providing us some spatial context. And so she also has a postdoctoral fellowship from the Canadian Institute of Health Research and has received some other awards for her graduate work. So I will turn it over to you, Miranda. Very excited to hear about your work.
Awesome. All right. Thanks, Lindsay. Let me just get my slides up. Okay. Yeah, so as Lindsay mentioned, I'm Miranda Hunter, and I'm a postdoc in the lab of Richard White at Sloan Kettering. And in our lab, we use a zebrafish model of transgenic BRAF V600E driven melanoma in order to study how tumors interact with neighboring tissues. So here at the top is an image of a fish from this melanoma model. And at the bottom, you can see a tank of these fish, and they're swimming around quite happily, despite the fact that most, if not all of them, have these large tumors. And there are many benefits to using the fish to study cancer. So they are cheap and easy to maintain in large numbers. They are genetically and pharmacologically tractable, and our zebrafish melanoma model closely recapitulates the human disease. So when I first joined Rich's lab, I became really interested in images that look like this. So this is an H&E stained cross-section through the adult fish. You can see where the tumor is because it's these kind of blackly pigmented cells that you can see just all over the place. And what I was really struck by was how invasive this tumor actually is. So I've labeled different regions where it's invaded into muscle and also where it's invaded into fat. And what this got me thinking about is how the tumor actually senses, contacts, and signals to neighboring tissues in order to facilitate the process of tumor invasion. So we decided to study this using a still relatively new technique called spatial transcriptomics. Oh, sorry, my images aren't appearing for some reason. Sorry, I'm just gonna stop for a second. I'm not sure why that's happening. Sorry, just give me 1 minute. The images are like pretty important, obviously. So I'm just gonna reopen the slides. Okay, let's try this again. Okay, so now that we can actually see the slides. So as I was saying, we decided to use a relatively new technique called spatial transcriptomics in order to look at how the tumor is interacting with the microenvironment. So here at the top, we have the fish that we used for our first spatial experiment. And I don't think you have to be a fish expert to see where the tumors are on these fish. And at the bottom, we have the actual H&E stained tissue sections from which we prepared libraries. And in all three cases, the tumor is this kind of purpley staining mass that you can see here. And I just want to take a second to highlight the power of the fish for these kinds of experiments, which is that, as you can see, an intact cross-section through the adult fish fits in its entirety on the Visium array without any need for dissection. So that means that we can study the genes expressed by the tumor, as well as the genes expressed by all of the adjacent tissues in their native environment. So I wanted to show some examples of what our spatial data actually looks like. So here on the left is one of our samples, and I've labeled some of the different tissue types present in this section. Now, what we can do is look at these spatial gene expression profiles for different marker genes of these tissues and see how well the spatial expression actually matches the histology. So for example, if we look at BRAF V600E, which is the human oncogene that we're using to drive tumor growth in the fish, we see this very high expression of BRAF, specifically where the tumor actually is. And we can repeat this for other markers of these different tissues. And we again see this really nice correlation between the spatial gene expression profiles and the actual histology of the tissue. So that tells us we're working with fairly high quality data. And spatial data can be treated like other types of transcriptomics data, like single cell RNA-seq data, where we can cluster the data points based on gene expression. But then with spatial data, we can map the cluster assignments back onto the tissue array and see how well the clustering actually kind of reconstructs the histology of the tissue. So that's exactly what I'm showing here. So at the top are 3 samples, and then the bottom we have the tissue arrays with each data point labeled based on its cluster assignment. And hopefully you can appreciate how well the clustering does in fact reconstruct the histology of the tissue. So for example, we have a large tumor cell cluster, which is labeled in this kind of like pinkish red color. And in all 3 cases, it localizes exactly to where the tumor actually is. And we see the same thing for the other microenvironment cell types present in the sample. But something we are really excited to see in this data was that in all 3 cases, there was a distinct cluster that localized exactly to where the tumor is contacting the neighboring tissue, which we have termed as the interface, meaning the tumor microenvironment interface. And it's labeled here in this turquoise color. So the reason this was so exciting to us is that if you remember, the original rationale for this experiment was to dig into the mechanisms by which tumors sense, contact, and signal to neighboring tissues. And the presence of this distinct cluster suggests that there's changes in gene expression occurring in this region that may be contributing to this process. So in light of this novel interface cell state that we uncovered in the fish, we were also curious if it could be conserved in human melanoma. So to investigate this, we analyzed a published human melanoma single cell data set, and we scored each cell in the data set for the relative expression of the human orthologs of the zebrafish genes that we found to be upregulated in the interface. And hopefully you can appreciate that in both the tumor as well as some of the microenvironment cell types in this data set, there were distinct subsets of cells that did upregulate these interface genes, suggesting that this interface cell state may in fact be conserved in human melanoma. So in light of this novel interface cell state that we've now uncovered in both the fish and in human data sets, obviously, the next question is, what are the genes being upregulated here and how could they contribute to melanoma invasion? So to begin to investigate this, I took 3 published human melanoma single cell data sets, as well as our own fish data. I identified putative interface cells in all of these data sets and then calculated the most highly upregulated genes in the interface. And I was struck by the fact that in all 3 of these data sets, the most highly upregulated gene in the interface is this chromatin modifier called HMGB2. And there isn't a lot known about HMGB2 in melanoma or in cancer in general, but we got some hints from looking at publicly available data sets. So from this TCGA data, we found that high levels of HMGB2 are prognostic in human melanoma. And when we downregulated HMGB2 with siRNA in a human melanoma cell line, we found that it significantly impaired the ability of the cells to invade in vitro. So kind of some hints that this protein may be doing something important in melanoma progression. But of course, but for me, I'm a cell biologist. So if I'm interested in a new like protein or gene, my first instinct is to just look at it in vivo and see what exactly it's doing. So I did exactly that. So I stained tissue sections from our zebrafish melanoma model for a marker of the tumor, which is shown here on the left, and then with an antibody labeling HMGB2 shown here in the center. And I'm again focusing on this interface region where hopefully you can appreciate that the tumor is kind of intercalating or invading into these adjacent muscle bundles. And when I looked at HMGB2 expression in these cells, I thought, all right, this doesn't actually really look that interesting. It looks like it's just kind of highly expressed in all of the tumor cells. But when I looked a bit more closely, I saw something that I did actually find interesting. Maybe put yourself on mute. So I saw something that I did find very interesting, which is that if I focus on this region of the interface here, although we see it everywhere, is that HMGB2 was only highly expressed in these kind of elongated or misshapen confined cells that are likely being subjected to high levels of mechanical force during invasion. So I've highlighted this in the images on the right. So we can see that I've labeled the more kind of elongated or misshapen cells here. And all of these tumor cells have very high levels of expression of HMGB2 protein. But then when we look at the more round or the more kind of like typically shaped cells, we see little to no expression of HMGB2 at all. So to me, this suggested that HMGB2 may in fact have a mechanosensitive role at the tumor microenvironment interface. So to look at this with slightly higher resolution in human cells in vitro, we decided to subject these human melanoma cells to mechanical force by growing them on these confined micropattern tracks. So here on the left, we have cells seated in an unconfined area, and we can see that the cells have adopted these kind of polygonal shapes that are representative of this cell line. And then in the images on the right, we can see here that I've seated the cells on these 5-micron diameter fibronectin coated tracks. And the cells are trying to kind of squeeze themselves into this narrow region. Now, when we look at HMGB2 levels in these cells, we can see that in the unconfined cells, we have high nuclear expression of this protein, which is what we would expect for something that modifies chromatin. But in the confined cells shown on the right, we can see that we now have the emergence of this cytoplasmic pool of the protein. And we think this is likely very similar or the same thing that we're seeing in vivo, and that together this supports a mechanosensitive role for HMGB2 at the tumor microenvironment interface, where it is likely modulating gene expression and chromatin architecture in response to the mechanical force exerted on these invading tumor cells. And these are all questions that I'll be following up on in the remainder of my postdoc. And then looking forward to my independent career, the topic of cancer mechanobiology is something that I plan to focus on in my independent role. And in particular, I plan to focus on the role of the mechanical microenvironment in cancer. And this is because the role of the tumor microenvironment, meaning the healthy cells and tissues that surround a tumor, has been appreciated for more than 100 years now. But the mechanical microenvironment, meaning the mechanical forces present across the tissue, has been studied much less, and it's just not clear how this could be contributing to tumor cell behavior. So in the initial years of my independent group, we will focus on tumor cell confinement during invasion, and we will study how confinement affects cell and nuclear shape, how it affects tumor progression, and how tumor cells sense and adapt to confinement. And looking ahead to the longer term, I plan to focus on other aspects of the mechanical microenvironment, including cell volume and tension and pressure, among other things. But overall, in my independent role, I plan to open up this new area of cancer cell biology, studying this role of this invisible but critical component of the microenvironment on tumor cell behavior. So finally, just to wrap up, I of course want to thank my supervisor, Richard White, and all of the members of our lab. And I definitely want to acknowledge our collaborators, Ruben Moncada and Itai Yanai at NYU, who are our essentially equal partners on the spatial work that I presented in the first part of my talk. And of course, I want to thank my funding sources and all of you for your attention. And I'm happy to take any questions. Thank you.
Thank you so much, Miranda, for a really exciting talk. So again, if you have any questions, please raise your hand or put them into the chat, but I will get started. So I had a question. So this finding about this mechanical, this confined feature is very exciting. my question is, can the cells sort of revert back? So have you actually tried that once a cell has been confined, have they been permanently, altered, or can they actually, is it more plastic in that they can go back to their unconfined gene expression programming?
Yeah, I think that's like a very important question. There's kind of some hints from the literature that this may in fact be I hesitate to use permanent, but a more kind of like lasting cell state, even after the force has been removed. One way that we're kind of planning to look into this, I'm just starting now, is we have a system where we can actually stretch the cells. And the way, we essentially are literally just growing the cells on like a PDMS membrane and then physically stretching them. But using that chamber, we can also like unstretch them. So I think, and that's something I plan to kind of just look at, gene expression and things like that, and see if we kind of like apply mechanical force, if we take it away, all the things that you mentioned, how that affects the cell kind of just generally.
Very cool. And then also in terms of this initial migratory property of these cells, you have all these nice spatial approaches to dissect, the changes at that interface. And so my question is, do you actually, what are the driving forces, do you think, for that? Can you actually read something out? Is there something with the gradients of factors that are secreted by cells in one side or the other? Or are you able to get that sort of resolution to figure out if there's any attractant forces, or what do you actually think is driving that initial metastasis or invasion?
Yeah, I think this is one of the biggest open questions. We have a lot of ideas about things being secreted. We have very little data to support this right now, but we've thought a lot about, could the tumor be secreting something? Could it be secreting even like mRNAs and like vesicles that could be taken up by the microenvironment? Could the microenvironment be secreting something? It's definitely possible. One thing that I didn't mention is that HMGB2 can also, there's at least evidence in the literature that HMGB2 can be secreted. So because we see it going to the cytoplasm upon confinement, we're thinking if this is kind of like the precursor to it being secreted and potentially like in an a amazing scenario, like being taken up by the microenvironment and kind of like modulating gene expression there. So these are all things that we're kind of looking into now.
Yeah, that would be fascinating.
Yeah.
We have one question in the chat from Rebecca. She's asking, could you provide any insight as to the protein content of the microenvironment at the interface of your tumors?
The protein content, we haven't looked at that at all. We have a lot of information on the mRNA content, of course, from our spatial data. Yeah, what we typically see is that the tumor is invading into muscle, which we think is just our zebrafish tumors seem to like invading into the muscle, and we see kind of typical muscle genes being expressed there. We are definitely interested in looking at kind of tumors invading into other cell types like fat or even interacting with skin cells. And if we see kind of a similar interface cell state being adopted there. So yeah, all things that we're definitely interested in.
Great. Well, thank you again, Miranda, for a really exciting talk. And we'll move on to our next speaker. So our next speaker is Jaye Gardiner, and she is coming to us from Fox Chase Cancer Center. And so today we're going to hear a bit about some of her exciting work studying cancer-associated fibroblasts. And Jaye has a number of fellowships. So she has received a Black in Cancer postdoctoral fellowship and also has been funded by ACS and a T32 fellowship. And also she spent a lot of, she's also very committed to science communications. So you might have heard of some of her work actually doing illustrations. So she's founded JKX Comics. So I hope that we'll be able to see some of her images that she's drawn in her presentation today. But with that, Jaye, I'm excited to hear about your work.
Thank you so much for that introduction, Lindsay, and thank you to everyone for being here today. I'm incredibly excited to tell you a little bit more about a recent project that I've been developing using bioinformatics that actually revealed a canonically endothelial cell signaling pathway, but in pancreatic cancer-associated fibroblasts. And the reason why my current lab is focusing on pancreatic cancer is because there is a dire need for new targets. So if you have 100 recently diagnosed patients only 11% of them will make it to 5 years after that diagnosis. And even still, those remaining 11% of patients will usually still succumb to the disease. So this is a very terrible disease that needs a lot more to increase our patient outcomes. Part of this, these abysmal outcomes, is due to the tumor microenvironment that you just heard a little bit about from Miranda's talk. But what's really special about pancreatic cancer, so on the left, I'm showing an image, a stained image from stained patient tissue, is that there's actually very little tumor cells that's shown here in orange. The majority is actually this tumor microenvironment, or TME, which for pancreatic cancer is predominantly these cancer-associated fibroblasts. So fibroblasts that have been activated into this constitutively active state that create a tumor promoting environment. And so these fibroblasts, or CAFs, also lead to a large excessive deposition of the extracellular matrix, so that it's very reminiscent to what's happening in wound healing or in fibrosis. Additionally, CAFs secrete a large amount of cytokines that lead to this immunosuppressive environment, and the density of the ECM that it secretes overall can lead to blood vessel collapse. So another really unique feature about pancreatic cancer is that there's very little new blood vessel growth or angiogenesis. All of this together, however, means that this disease is incredibly difficult to treat clinically. So the overarching question that we're looking in the lab is whether or not we can reprogram this microenvironment to selectively harness the natural tumor suppressive functions. And I say the natural tumor suppressive functions because ablating the TME in and of itself is a non-viable option. If we were to do so, this actually leads to more aggressive tumor growth. So the way that we do this is by really making sure that we have the ECM and can recapitulate this in vivo biology in vitro. And so by using patient-derived fibroblasts, we can actually culture our cells over the course of about a week so that they create their own cell-derived matrices and create a more three-dimensional-like structure. And by doing this, we actually found a large number of regulators of CAF function, the most recent of which, highlighted here, our 2021 study that I'll go into a little bit, forms the basis of my work. What's really interesting about this study is that we actually found a novel neuronal protein that is being specifically expressed during the disease state of pancreatic cancer. And when we did some in vitro studies knocking out this protein called netrin G1 and profiled the cytokines that are being secreted by these CAFs, we saw that the majority of these inflammatory cytokines are actually drastically decreased when compared to the non-knocked out control. If we look at the expression of netrin G1 specifically in the fibroblasts in patient tissues, we actually found that there's a variability in the amount of expression that is found in patients. And most importantly, that this differences in expression levels actually negatively correlates with overall patient survival. That is, the more netrin G1 that is being expressed in these CAFs or fibroblasts, the worse off these patients fare. So because of this, this really started to suggest from our in vivo and in vitro studies that the loss or absence of netrin G1 is starting to reprogram these CAFs like we're trying to do and create a more normal-like state. So because of this, my overarching questions then are, what is differentially being expressed in these netrin G1 high and low expressing CAFs? And if by doing these comparisons, can we find other key regulators of CAF function? So to do this, I worked to make lemonade out of these pandemic lemons. And so from the initial work from home period, I actually taught myself how to analyze bulk RNA-seq data that was publicly available. So I first set out looking for studies that used patient-derived pancreatic fibroblasts similar to what we use in the lab, and that also was looking at a comparison between CAFs and the normal-like fibroblasts, so that I could ensure that any differences with netrin G1 that I'm looking at is solely in the CAF population. And to just briefly go over what I did, just for the sake of time, I found two different studies that fit these criteria that I was then able to combine. After normalizing these data so that the only difference I could see within the cells was the difference between the CAFs and the normal-like fibroblasts, I actually was able to stratify and find Netrin G1 high and low containing CAFs. So just using these populations, so 2 cells in the high against 4 in the low, I then compared these for a differential gene analysis. Doing so revealed that 530 genes are different between these populations. So a large amount of genes are actually changing using Netrin G1 as a marker. The majority of which, so about 60% of them, are being downregulated in the Netrin G1 high expressing CAFs. To get a little bit more information about what these genes are doing or what pathways they represent, I did a clustered network analysis, and this actually showed that about 67 of these genes are already known to be highly interactive with one another or represent a number of pathways. So here I'm showing the clustered network analysis that shows about 16 different clusters in different colors. The largest or the most enriched pathway is shown here in red and actually relates to this TEC-related signaling. So this is something that's canonically found in endothelial cells, and specifically this kinase TEC was downregulated in the Netrin G1 high expressing cells, and its ligand that leads to activation of this kinase was upregulated, which may be due to compensation. Briefly overlaying this data, so the 530 genes that I found different with our Netrin G1 knockout RNA sequencing data, there are actually only 94 genes of which that were in common between the two. These 94 potentially representing Netrin G1 regulated genes, meaning that the remainder of this 436 could be novel Netrin G1 independent regulators of CAF function. If I overlay the 17 genes from that enriched cluster, so the one that had TEC and angiopoietin, the only 2 that are in common with our Netrin G1 knockdown are those 2 proteins themselves, suggesting that these are in fact something that is being changed when Netrin G1 is present, and because it's represented in our RNA-seq data, that they may be regulated by Netrin G1 themselves. But as I mentioned, these are canonically endothelial cell proteins, or they're most characteristically defined in endothelial cells. And so I needed to first kind of understand what it's doing there before I can start to interrogate what it's doing in the CAFs. So to provide you this greater context, TEC is a tyrosine receptor kinase that needs to dimerize for its function. And it has a variety of ligands. The most well-studied is angiopoietin-1, so the same ligand that I saw was upregulated in the public data set. But there are also 2 other family members in humans, angiopoietin-2 and 4, that can bind and activate the TEC kinase. When this kinase is activated, it leads to its autophosphorylation, and this then mediates downstream signaling that can promote proliferation, which is necessary for angiogenesis or new blood vessel growth with the endothelial cells, as well as endothelial cell migration and overall survival. However, there's also an additional function for TEC where it inhibits inflammation. And as we've already seen a little bit from the Netrin G1 study, that loss of Netrin G1 can change cytokines that are involved with inflammation. This is what I'm starting to look at first. So what TEC and angiopoietin-1 is doing in CAFs is currently unknown. And so the way that I'm going about to address this question is using the same 3D culturing model that we use to find Netrin G1 and our other CAF regulators. However, in addition to the matrix production period that I said occurs over the course of about a week, I will also be treating with various drug treatments or recombinant proteins, so either spiking in recombinant forms of the TEC ligand, so the various angiopoietins, to see if this is actually responsive in the CAFs, or an inhibitor. And after an additional 2-day period to generate conditioned media, I then collect all of my samples. So the media where I can use to profile for various inflammatory cytokines, specifically starting with IL-6 and IL-8, as well as the whole cell lysates for Western blot analysis, And what I'll be showing you today is for the phosphorylated form of TEC, which will be indicative of activation of the signaling pathway. So starting with the inhibitory study from an initial pilot experiment testing various time points, the turnover rate for turning off this kinase is incredibly fast. So I'm not showing you a Western blot for its activation here, but for the ELISA in both TEC inhibitor treated samples, it significantly increased the amount of these inflammatory cytokines being secreted. If we look at the converse experiment where I'm adding the ligands that should theoretically activate this kinase, I can see that compared to the untreated control, the addition of all 3 ligands leads to activation of TEC in our calves, as denoted by the darker banding for the phosphorylated form. And if we look at the inflammatory cytokines, very nicely for IL-6, we can see that activating TEC leads to a decrease of the IL-6 secretion, which this would actually be beneficial for patients. But this is currently inconclusive for the IL-8 cytokine. Collectively, this is starting to suggest that TEC may also be playing an anti-inflammatory role in our calves, akin to what's happening in endothelial cells naturally. So to just summarize what I've been showing you today, I used bioinformatics to find a distinct gene signature between Netrin G1 high expressing and low expressing calves. Further analysis showed that the most enriched pathway of this is TEC and angiopoietin, and that there is a core interconnected network of these genes, and that looking back at the biology of what it might be doing, that TEC and angiopoietin may have a novel role in calf function. So to wrap up this project, some things that I'm working on addressing now is seeing whether or not the calves secrete these angiopoietin ligands themselves. So seeing if there can be a cell autonomous regulation, or if they're simply responding to, say, the cancer cells or other things in the microenvironment. And additionally, if I want to use an orthotopic mouse model to see if constitutive activation of TEC in the Netrin G1 high expressing calves could abrogate tumor growth, similar to what loss of Netrin G1 does. So like most science, as you do more experiments, you open up a lot more questions than you do answers. And the one that I'm most excited about continuing to pursue in a little bit of a broader complex is what leads to the aberrant expression of these functional regulators. And I really want to, similar to Naomi, kind of harken back to what I did during my graduate career, which was actually in virology. And during the ongoing pandemic, we've seen a lot of evidence that viruses can really rewire the cells and have systemic effects in the body outside of the initial placement of infection. So building on what I did in my graduate career, I'd like to combine this with what I'm doing in the tumor microenvironment for my independent career, asking the overarching question, how does viral infection reprogram a cell to create a more fibrotic environment, which could be the predisposition to a lot of other diseases such as cancer. Additionally, as Lindsay had mentioned in my introduction, I've been involved with a lot of initiatives starting in my undergraduate career, actually, to build up the diversity, equity, and accessibility that is present in STEM. So be that through community initiatives, education, or through science communication, where I make a variety of comics and trading cards and illustrations like you saw throughout my talk. So what I would like to do is to build on everything that I've been doing so far and not just expand my science, but all of these initiatives for my future lab. So with that, I would like to acknowledge all of the members of the Sukerman Lab, especially my mentor, Dr. Etty Sukerman, who's just a really fantastic mentor and has taught me so much over these last, I guess, 5 years since I've started, as well as Doctors Francesconi and Benjamini Costa, who are the lead authors on that Next Gen G1 paper that formed the basis of my work and have mentored me a lot throughout my time here. I'd also like to thank all of my various funding and support, especially Leading Edge. That has been a really great community to be a part of as I start this transitionary period, and all of you for your attention. And I'd be happy to take any questions.
Thank you so much, Jay, for a really exciting talk and a lot of interesting future directions with this project. So we have a question in the chat from Carolyn Bartman. She wants to know, very cool, your data says that maybe angiopoietin can signal to the CAFs, but do you think maybe it could also act on endothelial cells to influence angiogenesis in the tumor?
Yeah, that's a really good question. So I feel like there's been very little studies on the endothelial cells themselves that are happening or that are there in the pancreatic tumor microenvironment, just because there's so little angiogenesis that is seen. I don't necessarily think that it's that angiogenesis can't occur, but just the density of the ECM that happens that really defines that tumor microenvironment. Anything that tries to start kind of gets collapsed. So it's certain that there could probably be a sort of effect that is there for the endothelial cells that are still present, but I don't think there will be something so substantial to allow for new blood vessel growth to occur.
Great. All right, I had a question about, I guess you found this in the pancreas CAFs, but do you think that this is, again, like cell type specific effects that you would see in different depending on the context of the tumor microenvironment? Has that been explored in CAFs very much with tech signaling?
Yeah, that's a really great question. Definitely nothing with tech so far and other fibroblasts. So I'd be really interested to see if other tumor microenvironments that might have larger ECM deposition or expansion of these CAFs kind of follow a similar phenotype, because then you can do some really neat comparative studies to kind of see what's turning them on and if it's consistent across all of these different contexts.
Yeah, very cool. And then do you know much about like the negative regulation of tech or these signaling pathways?
Yeah, so in endothelial cells, there's a related family member to TEC. So the protein name for TEC is Tie2. I just use TEC to differentiate between the other one, which is called Tie1. And that and another protein, PTPR beta, are the natural inhibitors of TEC that can respond to the different angiopoietin ligands. I actually looked for both of those in our fibroblasts, and neither of those are expressed. So it potentially, or at least I'll hypothesize right now, that Next-Gen might be filling in that role of the negative regulator. And so I definitely want to do some more studies to see if that pans out.
Yeah, that would be cool. One more quick question, and then we'll switch it over to our last speaker. So what mechanism this is from Ali, what mechanism tumor cells use to change fibroblast fate into CAFs? Are you considering looking at that in your work?
Yeah, so one of the main drivers for fibroblast activation, also known as myofibroblastic activation, is really just TGF-beta. So any cell that's in the microenvironment that is secreting TGF-beta will kind of activate a fibroblast. The neat thing about CAFs is kind of they're always turned on, and so they also start to pump out their own TGF-beta and can kind of have a field effect where the first ones that are converted, that are closest to the tumor cell, can then start to convert all of the other fibroblasts that are further away from the tumor. But with regards to if I want to look at that in my work, I absolutely want to do that for my own lab with viruses. So seeing if viral infection, even if it's not in the fibroblasts themselves, if it's the cell type that they are infecting, if that leads to changes that increase, say, TGF-beta secretion that could then alter these cells and kind of create a similar, more fibrotic-like environment.
Very cool. Well, thank you, Jay, again for a really beautiful talk. And we'll switch over to our last speaker. So our last speaker is Rebeca San Martin. So Rebeca, if you can share your slides. So Rebeca is joining us today from the University of Tennessee. And she is going to tell us today about her work. She's she has put a lot of effort into developing really sophisticated models to study prostate cancer biology and metastasis. And also she has a American Cancer Society postdoctoral fellowship. So we're very excited to hear about your work, Rebeca.
Thank you, Lindsay, for the kind introduction. So in thinking of an informative title, and I'm trying to see, can you see my slides?
We can, yes.
Okay. Next question, can you see my pointer?
Yes.
Okay, perfect. So in thinking of a title, I came to realize that, yes, I will be talking about the influence of the microenvironment in the genome architecture of prostate cancer. But I will amend that to a narrowed view of the influence of the calcium-rich microenvironment on the genome architecture of prostate cancer. So if I were to ask people on the street about a site of the body rich in calcium, most people would go directly to bone, right? And the reason that I'm interested in bone is because it's the preferential site of prostate cancer metastasis, and because it is the main reason of mortality in these patients. Now, why does prostate cancer goes to the bone is not well understood. But we have clues though, based on my previous work, which showed that protein content in the reactive bone microenvironment is very similar to that of the primary tumor. I believe that the microenvironment is an important homing and growth factor for these types of cells. My current work, it's focused on characterizing these microenvironment and how these, the presence of the microenvironment triggers changes in the genome organization of prostate cells, with the ultimate goal of characterizing the mechanisms by which the bone microenvironment influence prostate cancer metastasis biology. Now, the context of metastasis is very broad in prostate cancer because it involves the actual homing of the cells to the secondary site. And of course, the phenomenon of dormancy, where the cells arrive at the metastatic site and then stay quiescent, sometimes, sometimes for decades. But studying these events requires modeling a very complex environment. So throughout my career, as Lindsay kindly mentioned, I have developed several in vitro and in vivo systems to model bone biology and its many players. Like, for example, the calcified matrix and proteins associated to that matrix via calcium-coated surfaces, bone scaffolds that are humanized with human proteins and also human ECM. Now, to model the interactions with the cells of the bone, with the osteoblast, I have derived osteoblastic organoids that we can co-culture with your cancer cell of choice. And finally, to model the colonization, dormancy and progression effects, I have developed an in ovo, so in a chicken egg, in the chorioallantoic membrane of the fertilized chicken egg, xenograft system in which we can put all of these things together, from the matrix to the scaffold, from the scaffold to the organoid, into a compatible host. Bear in mind that although I devise all these systems for prostate cancer biology, they are by design very modular and thus usable on other cancers that also metastasize to bone like breast. But in the interest of time today, I will talk about my most recent work on amorphous calcium-coated surfaces. And this story begins with the COVID-19 pandemic, which when specialty plastics for cell culture, like, guess what, calcium-coated surfaces, went out of production. So I took a page out of the bone implant field and modified a system in which titanium serves as both the catalyst of amorphous calcium complex formation and also the surface for coating. Here I am showing you the chalky deposits that we observe on treated titanium and the structure of these calcium surfaces on scanning electron microscopy. Now, the biggest disadvantage of this system for experimental work is that it's not permeable to light, and thus it's impossible to see cells under the microscope. So we modified this system to coat plastic vessels that we commonly use in cell culture. And dare I say, we were kind of successful in doing so. But interestingly, it should come to no surprise, that prostate cancer cell lines that were derived from bone metastasis really like the calcium matrix in titanium or in plastic. They attach better, they grow faster, and they also can live in these surfaces without testosterone. And this is important because one of the first lines of defense that we have against prostate cancer is ablation of testosterone, is removing the testosterone from the equation, and this being a male-derived tissue, it actually gets quiescent in the absence of testosterone, and my calcium surfaces can bypass that completely. What is very surprising is that early adenocarcinoma cells behave the same way. So they also grow faster. They really like these surfaces, and they can survive in the absence of testosterone. So an early carcinogenic event actually survives really well in the calcium matrix, and this will become relevant in a second, I promise. So now I can use this system to query how the presence of calcium can affect the genome architecture of cancer cells, as I previously mentioned. And this is important because it is estimated that 1/4 of all patients diagnosed with early prostate cancer already present with circulating tumor cells. This means that these cells could potentially arrive at the bone niche through the bloodstream, and that from then on, their biology would be informed exclusively by the bone microenvironment. So in order to investigate the genome architecture, we use a technique that is called chromosome conformation capture, or HiC. HiC is a sequencing-based technique in which the interactions between genomic locations can be visualized in a heat map that looks like this, and with the chromosomal coordinates plotted in the X and Y axis, and the intensity of the color denoting a frequency of interaction. That means that, for example, this loci with itself interacts greatly, while it doesn't interact at all with this region. From this type of data, we can derive information as to which areas of the chromatin are open to transcription and which are closed or silenced, and we call these areas compartments. Here, I am showing you two hypothetical conditions where the normal cell is on the left and the cancer cell is on the right, or the untreated and the treated. As a cell progresses to malignancy, it is possible for a region of the genome to switch compartments from open to closed, and that results in a bigger closed compartment. The result of this switch could mean that the genes that are at that location are silenced, which would be catastrophic if these genes were to function as tumor suppressors. Conversely, a region of the genome could switch from the closed to the open conformation, resulting in this bigger green area or open compartment. And likewise, this would have terrible consequences if the genes activated by these switches were oncogenes. But in order for me to investigate whether things switch from open to closed and so on treatment or on progression, I realized that I did not know what the genome architecture of a normal epithelial prostate cell line looked like. And to make matter worse, we only had low resolution data for adenocarcinoma cell lines and no information at all about metastatic cell lines. So we set up to analyze the genome architecture of a cohort of 9 cell lines that are the most widely used models for prostate cancer, to set up the baseline for any inductive experiments. And we recently published this in JCV. To my surprise, most of the genome compartmentalization across cell lines remained constant, regardless of the disease stage. So if I were to zoom to this region, for example, I want to draw your attention to the intermediate regions where the structure looks exactly the same. However, in these areas that are highlighted by the squares, it is clear to see that there are regions, specific regions of the genome, that actually change with progression. So from the normal cell line, in this case, for example, this region is consistently in the closed conformation and starts to become open, if you will, in the cancerous conformation, and it gets aggravated in bone metastatic cancer. In total, of these genes that follow that type of consistent change and progression, we only observe 402 genes, out of which 360 genes switch from the closed conformation to the open conformation. And what's really interesting is that 80% of those genes actually cluster. They are in proximal clusters, they are neighbors to each other, suggesting that maybe the transcriptional changes of induced genes can actually make the whole region switch into an open conformation, affecting adjacent passenger genes. So further, when we do nearest neighbor analysis of compartment signature genome-wide, we observe that the cells actually segregate in a very logical, if you will, progression, genomic progression from the normal cell model to the early cancer and to three distinct arms of metastasis. These are atypical and traditionally believed to be androgen independent, but these two arms are actually bone metastatic. And what makes it interesting is that the cells segregate based on their racial origin. So these 3 cells are of African American origin, while these cells are of European origin, suggesting that these patients have a divergent oncogenic pathway. And we characterize this just based on conformation of the genome. Now, I would like to talk to you about a fascinating phenomenon observed in the bone metastatic cells, because now we can narrow our query to these genes that both change compartments and transcription. And more importantly, now I can associate the RNA expression level to a genome architecture status, albeit indirectly. So in this particular cell line, the switch from the B to the A compartment has already occurred. So in the control conditions, these genes, which come from that massive list of genes that we compiled, we identified as switching compartments, are already in the active form. Bone metastatic cell lines growing in calcium surfaces behave very different than those grown in normal cell culture plastics. So I'm trying to bring back that microenvironment factor into the genome organization. This data, I would like to draw your attention to this gene, which encodes for prostate-specific antigen, which is the primary diagnostic test we use continuously to monitor our patients. So if we take the cells in normal control, in control conditions grown in plastic, it is clear to see that for these genes, the transcriptomic profiling changes just by the presence of calcium. More importantly, some genes become refractory to the absence of testosterone in the presence of calcium. However, kallikrein-3 PSA in this cell line, which is of an European origin, is a really good indicator of a disease that would be present in a testosterone condition enriched with calcium. However, that is not the case for cells of African American descent. Our chemical castration model, if you will, on plastic results in transcriptional induction of this marker, while the presence of calcium abrogates it completely, even in the presence of testosterone. The grave implication being that these patients are underdiagnosed with metastatic prostate cancer because, as I mentioned before, their pathway to oncogenesis is different. Overall, these results show the importance of incorporating biologically relevant models in the research of prostate cancer microenvironment and its complex interactions, and of course, the need for better development of diagnostics and therapeutics. My career path has been very diverse and long, but suffice to say that I am a biochemist, chemist, and cellular molecular biology by training, who specializes in reactive microenvironment and cancer progression. And I foresee my independent group working in bone metastasis, colonization, dormancy, and progression with a focus in working in diagnostics and prevention that actually represents treatment and diagnostics for Latino, Indigenous, and African American populations. I would like to thank my mentors, my funding, of course, the leading edge, and I would like to entertain your questions.
Thank you so much, Rebecca, for a really beautiful talk. So again, if anyone has any questions, please raise your hand or put them in the chat, but I will start. So what do you have any hypotheses about what this actual mechanism might be of how calcium is regulating PSA, or and how that might be different in the two populations?
Yes, we have queried for a number of calcium transporters and downstream effectors of calcium exclusive metabolism. Calcium is also a very important player in melanoma. So the S100 family of proteins that induce alternative signaling. We know that in the presence of the calcium-enriched surfaces, the signal of androgen receptor binding to its own targets is enhanced, suggesting that androgen receptor doesn't care about the presence of testosterone anymore. So we can just, we induced, like for example, in the African American cell lines, we can induce PSA production independent of androgen just with the calcium, with an AR occupancy in those genes. So yes, we have ideas.
Very, very cool.
Thank you.
Any questions, any other questions? So I guess another, so do you think that this, obviously androgen receptor is very important in the chromatin remodeling, because you have this really nice feature of how these like local neighborhoods are sort of changing. Do you know what the actual, like if there is any hints of a chromatin related mechanisms that's driving, because it doesn't seem like everything's changing. You have these very specific neighborhoods that are kind of changing.
Correct. Yes, that is a really relevant question. So calcium is not the only game in town. We are currently modifying the calcium surface with the reactive protein profiles that we observed in the bone metastatic site, such as tenascin C and collagen 1, because those proteins also play a very important role in the informing of the nucleus, right? But yes, there are very relevant chromatin modifications that happen. But right now, what it looks like is that these cells are reading the signaling in the environment and transcriptionally changing a driver gene that is needed to survive under those conditions. And then, unfortunately, it has very close neighbors. And the very close neighbors are the nasty oncogenic signaling that comes in play because you try to survive by making the one gene. So it's in my view right now, my hypothesis is that we have driver genes and neighbors that come along. Yeah, that's a really relevant question.
Awesome. All right, we have a question from Jay. So apologies if I missed this at the end, but do you have any insights into what would be better markers that would work for all men? Your work is fascinating and very important.
Oh, thank you. I would like to think so. So the answer is no. Unfortunately, it's early days, right? There has been we know that African-American men and Latino men suffer from very aggressive prostate cancers that go underdiagnosed. We know that they are accompanied by very, very aggressive microenvironmental signatures. But the fact of the matter is that this is a very understudied field, right? So, for example, the African-American cancer metastatic cells that I am working with were derived in the last decade or so. So if we don't have the appropriate tools to query the biology, it's very hard to query the biology. So we have ideas. Out of these 240 genes, there's a subset of genes that change consistently in every single patient or in every single cell line, regardless of racial origin. So I would advocate to any of those that we can query through blood testing. But I know, at least I feel that PSA is not one of those.
All right, great. Well, thank you again, Rebecca, for a really exciting talk. And I just want to thank all four of the speakers. It was very impressive a group today. I learned a lot about all of your work. So very exciting. I just want to close by just reminding everyone that thank you for joining us today live, but we also will have these talks recorded and at some point will be available on the Genelia website, so you can check them out there. And please remember that this is an ongoing series. So we have every Monday, Wednesday, and Friday at noon across the month of July. You can check out other leading edge fellows in different fields. So thank you again for joining us today, and I'm looking forward to seeing you soon at the next one.